Welcome back, guys. So yesterday you should have looked at our anchor phenomenon video for Unit 2 and written your CER. If you haven't done that yet, please go back to yesterday's post and make sure you do that. So this unit that we're talking about now, we're going to be starting with, I guess not so much starting, but continuing with this idea of cells and how they divide. And we're going to move on to how we get from DNA to a full organism. So by the end of this unit, you should be able to answer the question exactly of why siblings look different from one another, including the cats that we saw in our anchor phenomenon video yesterday. So today we are going to start with cell division. Okay, so why do cells divide in the first place? So why can't they just grow forever big? So it's something I want you to think about for a minute, because we know that like the cells of your body, they're super, super tiny. I don't know if you've seen blood cells or cheek cells under a microscope before, but you need a microscope to see individual cells. So we know they're small. Okay, so the two main reasons why cells divide rather than growing larger. So the larger a cell becomes, the more demands are placed on the DNA. So the way I think of this, let's say there is one particular cookbook, right? Like it's Thanksgiving dinner and your grandma has everything she cooks in that one cookbook. Well, if it's just her cooking, it's not a big deal. She can flip to whatever page she wants and make what she needs. If there's like grandma and your mom and like 5 of your aunts all cooking at the same time, that cookbook is under a lot of strain. It's hard to get the instructions you need. So like a cookbook, the nucleus of your cells has a whole bunch of DNA in it, but the more things I'm trying to make out of that set of instructions at the same time, the more difficult it is. Okay, so a bigger cell is going to need to make more things at the same time. So it's very inefficient for our nucleus. So the second reason is that if the cell grows too large, it has trouble moving nutrients and wastes across the membrane. Things can only diffuse so fast. So I'm going to tell you right now, you're going to want to take notes on this slide. I know this is going to be on your cell division test, okay? This unit's going to be pretty big, so there's going to be several quizzes throughout this big unit, and the first one is going to ask you about this, so just be prepared. Okay, so our DNA has limits. All of the information that a cell needs to function is stored in the DNA, which we know that already. We talked about that in our cell analogy, that's what a nucleus did. So DNA is packaged into chromosomes. So what this vocabulary term means is that it's just a long piece of DNA with, it could be thousands of genes on it. So each gene has instructions for a protein. That's why we call it a cookbook. There's lots of recipes for proteins. That's all a gene is. So for sickle cell anemia, there is a gene that codes to make hemoglobin. Well, if you have a typo in that recipe, you make the wrong protein. So that's why people have illnesses when they have sickle cell. So the cell is constantly making copies of these genes and sending copies in the form of RNA out to the ribosomes. So I'm going to interrupt myself for just a second. I know there's a lot of stuff underlined. This PowerPoint was originally set to go with guided notes, which we're not really doing this semester. So don't worry about the randomly underlined terms. It made a lot more sense when I taught this live, okay? So just write down what you think is important. Anyway, getting back to what we're talking about. So later in this unit, we're going to talk more about how this process works, though, how we go about going from DNA to making proteins. For right now, we're not super concerned with this. Our main idea right here, though, is just that if you're making too many proteins at the same time, we can't access the DNA enough to make that happen. So that's, as it says on the slide, there's something of an information crisis where the DNA cannot keep up with the demands the cell's placing on it. So this is one of the reasons why cells stay small, is so that there's a reasonable amount of limits. or a reasonable amount of demands made on it. I kind of look at it like classroom size, right? If there's too many students in a room, it's really hard for a teacher to meet all of the needs, all the things that those students need, okay? All right, so growing cells need more food. So a cell has to take in, let me move my face, guys. Is to take in a constant inflow of food, oxygen, and water across the cell membrane. So waste products also have to be diffused out of the cell. A cell can't just go to the bathroom and go pee. It can't do that. It has to diffuse all of that waste out through the cell membrane. So a large cell requires more food and water, but it's also making more waste. So as the cell grows, the volume, the amount of stuff inside of it increases far faster than the surface area or the outer area available to do diffusion. So what happens, we see in this picture, right? It's a little hard to see, but if a cell is 1 nanometer on each side, its surface area is 6 nanometers squared, but its volume is only 1 cubic millimeter. So it's 6 to 1, right? We only go up 1 nanometer, right? One more. So then our surface area is 24, but our volume is 8. Okay, so we went from 6 to 1 to, what's that? 24 divided by, it's 3 to 1. It cut in half, okay? So then we have 4 on each side. So surface area, we basically add up the area of each side. So 4 times 4 is 16. And we're going to need to multiply that by 6 because there's 6 sides on each cube. I don't know if it's going to give me that math. But anyway, we can see from the first two that that's a big jump. So the cell can't get enough food in and it can't get enough waste out. So cell division then is where a cell, it grew, but now it's breaking in half basically. And this picture, it looks kind of gross, but remember that real cells, they look kind of gross. So these booger looking things, those are cells, and we can tell they're almost finished with cell division. They're still connected just a little bit though. So one mother cell will make 2 daughter cells, and the 2 daughter cells are identical to each other and to the mother cell. This is another thing that's going to be on your quiz. You're going to need to know like basic cell division, like mitosis. We have 2 identical daughter cells. We're going to learn that there's another division process used to make sex cells. It's a whole different thing. So just try to keep it straight. Cell division or mitosis, 2 identical daughter cells. Okay, so each daughter cell is half the size of the parent, but immediately begins growing. So here's another microscopic image of cells dividing. And as we see, they're about done. So a typical human cell has about 2 meters of DNA. That's a bit over 2 yards. So before the cell can divide, all of this DNA must be copied. So we know that a cell's microscopic, so its nucleus is even smaller, and it has all these tiny, tiny, DNA strands wound up to make our chromosomes. But if you unwind it, it's actually 2 whole meters, which is kind of crazy. But each daughter cell needs a complete set of all those instructions. So each species has a characteristic number of chromosomes in each cell. This cuts off at the bottom of the screen. Humans have 23 pairs of chromosomes. So 46 total. That's just a thing about us as people. So each cell must first copy its chromosomes before cell division occurs. So each daughter cell can have a complete copy of that information. So the actual cell division part happens in 2 main stages: mitosis, or the division of the nucleus, and then cytokinesis, which is the division of the cytoplasm. That's another thing going to be on your quiz, okay? Mitosis divides the nucleus, cytokinesis divides the cytoplasm. So the chromosomes are only visible during cell division because they have to coil them all up. Like when you're trying to put away your controllers for a video game console, or when you're trying to put away Christmas lights. It's a big mess if you just put them loose into a box. But if you wind them up, it's a lot easier to divide them up and store them. So that's why our DNA does that, so it doesn't get tangled and lost. So this is a replicated set of chromosomes. So there's 2 sister chromatids, each side is a chromatid. And then this little part in the middle where they are united is called the centromere. So this is actually a double set of chromosomes here. It's ready for division. All right, if you're writing notes, leave you a second. Okay, so the cell cycle. So this is what I want to focus on today. We will talk more in depth about the steps of mitosis in the next video, but let's talk about the cell cycle as a whole real quick. So the cell cycle is a series of events that cells go through as they divide. This is the entire life cycle of a cell here. So the cell cycle is the time from it's formed until the time that it divides. So during the cell cycle, it grows, it prepares for division, and then it divides to form 2 daughter cells. All right, so the cell cycle has 5 major phases, and you're going to need this for your assignment today. So there's G1, or the first gap, S, or synthesis, G2, or the second gap, and then mitosis and cytokinesis. So this is the kind of information you're going to need for your activity. You're going to be labeling a diagram and answering some questions about it. So make sure that you are taking good notes or that you know where the slide is, because you're going to need it. Okay, so before a cell can begin mitosis and actually divide, it has to do 2 things. It has to duplicate all of its chromosomes so that each cell has a complete set. And then it has to produce enough organelles for the 2 daughter cells. So these preparations happen during gap 1S and gap 2. So these are collectively known as interphase. I'm going to show you in a minute. This is why there's 3 interphase labels on your activity, because around here, this arrow around G1S and G2, all of that is interphase. It's what happens between division steps. Okay, so in G1, the cell doubles in size, and the enzymes, the organelles, and other molecules are going to double in number. So S phase, this is where DNA replication happens. That's another thing that's going to be on your quiz. So write that down. So G2, the cell assembles the special structures needed for cell division. So it's not as simple as just ripping yourself in half. There are specific organelles and structures the cell needs in order to do this successfully. We talked about centrioles a little bit in the organelle lesson, but those were like the reels that a cell needs to split its chromosomes, or the sister chromatids, in half. It also needs to make these little like protein like fishing lines that attach to that centriole so that it can reel the sister chromatids apart. So when interphase is complete, the cell is ready to begin cell division. Okay, so this is actually where we're going to stop our notes for today. All right, we got through interphase, and we know that mitosis and cytokinesis are next. We're going to talk about that in the next lesson, but I want to show you your activity for today. So let's get out of this. All right, so this was originally a color sheet that I handed to students. So what you're going to do is you're going to open this with Cami. I'll give it a second. So I know that some of this is going to be a little difficult to understand until you have our next set of notes. That's fine, which is why even though it says it's due today, if you turn it in after the lesson on Monday, I'm okay with it, but we should be able to figure most of this out, okay? So on this diagram, they give you a word bank of labels here. So label the diagram with the following labels. and then we can color it. So we've got a text box here. Let's say we want to put G1. So G1 is going to be this over here. So draw our little, sorry, it's early. So G1. Is that how they put it? Yeah, G1. Cell grows. Cool, so we did it. And so then it says for G1, we should color it blue. So is it gonna let me, what tool should I use for that? Drawing, no. All right, we can try this. So I'll get a good blue here. Oh, that's ugly. Guys, why'd I do that to myself? Cool. Drawing, we want this thicker. We also want it blue. Okay, so we select blue because it says that gap 1 is supposed to be blue, right? So it says lightly color gap 1 blue. So I can just color it blue. Oh, we can even see our text. Cool, cool. It's okay if it's not tidy, the tidiest in the world, but just, you know. All right. So we just go through and we put the labels where they go, and then we are going to color it according to the directions here. And, you know, you can color code it for yourself if that's going to help you remember. S-phase should be yellow. G2 is supposed to be red. So like wherever you see those, or wherever you put that label, you're supposed to color it that. It says color the part of the arrow indicating mitosis purple. And then the part of the arrow indicating cytokinesis yellow. So I'm going to show you that right now because this is the #1 part where students mess up. So mitosis, 1, 2, 3, 4 stages of mitosis. That's this right here. The part of the arrow for that. And then the part of the arrow for cytokinesis is yellow. We've got like a little LSU arrow going on here. I know it's not tidy. I'm just trying to show you what to do. So then down here, a lot of this we talked about already. Okay, so what is a series of events cells go through and divide? That's a cell cycle, so we talked about that. The longest stage of the cell cycle, guys, it's interphase. All right, so pretty much all of this you should be able to answer. The steps of mitosis we don't know, just a preview for tomorrow. I'm gonna put it on here, so those of you watching this video, you're gonna be on top of the ball. That's totally not English, I'm so sorry. Okay, so there's this acronym PMAT because I know you gotta do this. So PMAT, prophase, metaphase, anaphase, telophase. I don't even think, do you have the labels on there? Yeah, I think you got, yeah, you got all the labels on there. So that's the order that the stages of mitosis go in, okay? Please let me know if y'all have any questions, and I'll see you guys in the next video. Take care.